So wake up. There will be a very interesting session. We will talk about the systemic interpretation of the large amount of genomic data with the aim to translate molecular alterations into the clinical indications. And that could be done with the help of molecular tumor boards. And I'm very pleased to introduce the first speaker of today, and also my co-chair, Martin Zoche. Martin is a director of the Molecular Tumor Profiling Department of Pathology and Molecular Pathology in the University Hospital of Zurich. He's also a lecturer in the University of Zurich. But there's another thing which bonds Martin to Latvia, and that's additional peculiarity. Ancestors of Martin's family from the wife's side, in fact, inhabited the Schlockenbecker Palace in 16th century. And the son of that ancestor was actually the last bishop of Cesis until 1621. So welcome back, Martin. So the floor is yours.
Thank you very much for this very nice and kind introduction. Yes, we have some relatives 400 years ago which immigrated to Latvia and wanted to make their fortune life here in this Baltic region. Coming back to molecular tumor boards, and today I would like to introduce you how we have set up the molecular tumor boards at our university hospital in Zurich. And I want to start with a slide which shows the development actually of precision medicine from just 20 years ago to now. 20 years ago, we had for lung cancer, just as an example, we had for lung cancer just more or less a few chemotherapies available to treat all patients. And if we are looking now to 2020, a lot of different molecular alterations can be tackled and treated in a very personalized way. And even immunotherapy is not on here on the slide, and immunotherapy is definitely one of the most important therapies nowadays for lung cancer patients. Just to give you an overview and keep you back reminded on this early morning about how much advance actually we have done in the last 20 years in cancer. And of course, with all these different drugs which are available nowadays for tumor patients, we have a huge amount of different tests which can be given to the patient's biopsy. And that means really the pathologists, the molecular pathologists nowadays really have to decide and to discuss together with the oncologist which tests are useful in which area. And you can see it in some numbers. So these are just a few pictures of our laboratory. But you can see here on some numbers that the amount of molecular tests over the last years really more than doubled. And nowadays we are starting with comprehensive panel tests. And these comprehensive panel tests are more or less done for almost every metastatic patient. And of course, if you set up such a molecular testing, it's sometimes difficult in the pathology or in the oncology to decide which test has to be done when. And we have set up testing algorithms together with the oncologists, with the pathologists, to decide that every patient really gets the same way of getting diagnosed with tumor. And here on the left side, you have the example of an NSCLC stage 3B4. So after the diagnosis, which is really the classical setup of a pathology, H&E staining, IHCs, the pathologist decides if this is an NSCLC or a CRCLC, then the decision is taken about adenocarcinomas, squamous cell carcinomas, and the adenocarcinomas, which are the majority of lung tumors, then are going into an algorithm of molecular testing. So we do a fast testing with EDILA for BRAF, KRAS, and EGFR, and the results are already there within 1 or 2 days. So that means for advanced cancer patients, these patients can be treated if these tests are positive within really a few days. We are saying the patient should leave the hospital already with a therapy. Then we do additional IHCs, ALK, ROS1, PD-1, PD-L1, and at the same time we are starting the foundation test. That means a comprehensive panel test with more than 300 genes to identify what are the underlying mutations in the tumor. And then the results are there after 2 weeks, and then all the results together are going into a tumor board and discussed together with the oncologist there. We have the same for pancreatic cancer. Don't want to go into detail here, but also a comprehensive panel test is in the center, and in the end, the results are discussed in the tumor board. So how the tumor board looks like, and I think we have already seen yesterday, and this does not differ from other tumor boards in other hospitals. Essentially, you have 4 or 5 different expertises in the tumor board. So you have, let's start with a clinical oncologist. So you have the physician who is treating the patient and knows best the patient history. You have the pathologist there. We have people from the genomics and bioinformatics, molecular biologists, and we also adding clinical geneticists, that meaning human geneticists, who can advise later on the patient on germline mutations. But when we are coming together in the tumor board, some challenges came up. So the amount of data we are discussing and the time per patient available is really a topic. So that means all the information which are needed in the tumor board has to be present at the time. So you cannot go into a database and start looking at something when you discuss a patient. There are so many experts coming together, time is really valuable. That means everything has to be on the spot available. We have different standards for reporting. Every pathology, every test is reported differently, the molecular alterations, and this is really a topic in terms of when you want to make them visual, when you want really to rate which are pathogenic mutations, benign mutations. So therefore, you really need a standardized reporting system. Of course, limited facilities. We have a lot of people who are joining from the outside, external oncologists from other hospitals, and they have to be integrated into the tumor board. And there are different aspects. One aspect is a technical one. I think nowadays this can be solved with the COVID pandemic, that everything is on Zoom. So we have set up a system here, even though the system has to be a safe system because we are discussing patient-sensitive data. That means that this can be not just a Zoom where everything is recorded by Google and other companies and can be stored somewhere else. So we need a safe, integrated system. The clinical trials is another topic. We're always discussing if a patient can join a clinical trial. And here we need the information, and the information always is not available in which part of Switzerland a clinical trial is open and can be used for this patient. So we need this information as well. And of course, limited access to drugs targeting the genomic alterations, but this then comes a little bit later. So here the oncologist really has to discuss with the pharma company and the health insurances how to come up with it. So what we need more visualization and all data available in one tool. So we have set up this tool, and here are my 2 colleagues, Fabian and Abdullah, who have essentially programmed this tool and further developed over the years. We have published this, so this tool is public available, and we have created a browser-based software which is available on every hospital computer. So that means every oncologist, every physician in the hospital can enter into that tool. We have a fully automated annotations. It's interactive, and the goal is that we can visualize all the NGS results. And here we are talking easily about 10, 20 pathogenic mutations we identify for a tumor patient. The software, we called it MTB Pilot, is just one part of a whole suite of softwares in my department. So it starts with a LIMS system where all the raw data and all the lab data are going into. These data then are stored in a database together with the results, and here we are interacting with other systems in the hospital, for example, the overarching pathology system. And we have, for the MTB Pilot, for the molecular tumor board software, created a cloud. That means there is a solution that every physician can enter with name of the patient and birth date, the information for this specific patient. And we are storing all alterations, genomic alterations, in a special format. We call this format OAPL, one alteration per line file format. And this is shown here on the next page. This format really gives all the most important information. We have a lot of bioinformatic file formats. We have the variant call file, the BAM file, and others. But this information in these files is not sufficient for getting all the information ready for the tumor board. For example, we have to know if the functional impact, is this a known mutation, a pathogenic mutation? Is it a variant of unknown significance? Is it a benign mutation? These things we have to know, we have to filter, and this is not provided by a variant call file, for example. And therefore, we have created our own format, because there is no international standard available. So the National Cancer Institute have a mutation annotation format, which is quite similar to what we have created, but there are still some differences, and we have not agreed and harmonized yet internationally. The software I already mentioned is fed by information from our database as well as other systems from the hospital. But also we have a lot of information we are generating and can present from public databases from the outside. And everything which is fed into the software, and this is, we will go into detail, this is a view of the software, the surface. The other databases which we are adding here are added instantly, on time. That means at that time we need the information. So, the software looks into the database and creates from the database the information and reflects it in our software. So that is how the surface looks like. So here we have the patient selection. So we have many patients discussed in the tumor board, typically between 5 to 20, sometimes even 25 patients in 1 week. So we are doing the tumor board every week on Thursday, 2 o'clock. This is a holy date for all oncologists and pathologists at the hospital, where everybody shows up for the tumor board, for the molecular tumor board. Then we are clicking and choosing one patient. Then the patient information is shown, and also we are showing all the other tests which are already done for the patient, so that we can see already the history of molecular tumor profiles and can click between the different tests to understand better is there which mutation developed over time, which mutation may be diminished, so that we get a good overview. And then we have additional information. So we are looking into the biomarkers, TMB, MSI, LOH. We have an ideogram we can show on the different chromosomes, which chromosomes are affected by the tumor alterations. And then we have down here in detail information about the somatic and germline mutations. Additional resources we can show, of course, we can look into the report, and also we can look into slides, H&E slides. We can show directly if there's a question, is it really the tumor we are discussing, the pathologist wants to see again the morphology, then we can look into this data. So we are showing here the short variants. We are showing copy number alterations and rearrangements. These are the most three important informations, and all these information is shown here at one spot and is visual, and these are visualized. Also, we can show the therapies, that means the drugs, which are eligible for these different mutations, as well as all clinical trials around the world. So we have a Google map, and we can switch to the clinical trial section here, and then we can show all clinical trials which are available in Zurich, in Geneva, in Lausanne, in the area around, but also in wider. We can look to France, to Germany, to Italy, and discuss if the patient might like to travel also to another country, to Milano, for example. Many patients from Zurich are going for clinical trials to Milano. To get a little bit more information about the features from the SNV panel, so for example, when we are clicking here on the molecular viewer, we can show the 3D structure of the protein, and in the 3D structure we are visualizing exactly the mutation which is shown up from this specific patient. And this is important information, because then we can look, especially if the mutation is not known, if it's a variant of unknown significance, because then we can decide if this is a mutation in a domain. This might have a higher impact than having a mutation maybe in the N or C terminus. We also can look into databases. Here is an example from TCGA. And here we can look how often this mutation arises in the database. We have a domain viewer and can see in which domain the mutation occurred. A fusion viewer is also integrated. That means we can visualize a fusion product and simply decide on this information if the fusion product is functional. This is important information if you think, for example, about NTRK fusions. The C-terminal domain is a kinase domain of the NTRK fusion, and the N-terminal domains are the regulatory domains. And if the regulatory domains are diminished, then you have a functional NTRK domain, a functional kinase domain, which can be not regulated anymore. And this has to be identified because, for example, if you have a shift in the triplet sequence and you cannot have a functional NTRK kinase domain anymore, then of course the patient is also not eligible anymore for NTRK drugs, larotrectinib, entrectinib. These decisions we can do here in the tumor board when we are looking into the fusion products. The software is available online, so you can access it with the domain mtbpilot.org. You can put up your own data, you can play around a little bit. Maybe it's not possible to use it in the tumor board because not all functionalities can be set up in the public version, but at least you can see. We also have a Swiss molecular tumor board, which is by the SPHN initiative, Swiss Personalized Health Network. So we are discussing once per month with other universities, very complicated cases, or that we are looking into interesting cases just for training purposes, that we want to inform each other about very interesting mutations we have identified. This just is my last slide, giving you an outlook what will be possible in future, because the Holy Grail still is linking all the genomic alterations which we have already in databases. This is easy to maintain. You have seen the OAPL format. It's very easy to put these information into databases. But it's much, much more difficult to put in all the clinical patient data into a database. What was a treatment after we have done the tumor board? What recommendation we have given from the tumor board has to be captured. Then we have to look into the therapy, which therapy in the end was really given and decided by the oncologist. What was the outcome of the therapy? Was it positive for the patient? Was it negative? And we are now in an early stage programming a database where we can capture also all the clinical information of the patient. And this will be the future. This is just the first glimpse on it, so that we can see the complete patient on one page digitalized and all the information are available, and if you need more detailed information, you simply click on these different bars here, and then you see very detailed what information stays behind this, and then you can discuss this information in a tumor board. Yeah, again, the slide, everything is only possible with a dedicated and motivated team. So once per year, we are going actually in this Ābols, doing a retreat, science and hike, we call it. 2 days we are working, 2 days we are having fun. And yeah, with that, I want to conclude. Thank you.
Thank you, Martin. I invite any questions. And while I don't see anybody coming to the stage, I will start with myself. I think it was Lars who mentioned yesterday, who referred yesterday to American Genome Project, which was launched in 2010, and actually demonstrated the significant difference in the spectrum of childhood cancers compared to adult cancers, and also the large genomic differences among the childhood cancers. Up till now, the comprehensive molecular genetic data analysis in pediatric cancer has been limited because the patient numbers are small. On the other hand, we heard yesterday from Rob Peters, who is a medical head of the biggest pediatric cancer center in Netherlands, that they run 3 molecular tumor boards per week. Clearly, countries like ours, we have like 10 times less patients than Netherlands. So what would be advice and what would be way out for the countries like ours? How should we approach? Because the knowledge is very, very specific on a specific type of the cancers.
Regarding the tumor board, I think there's no limitation. If you only discuss 1 or 2 cases, you can go after 15 minutes maybe, and not 1.5 or 2 hours. What in the end, what you need is that all the experts are available. And I think this is really the critical point, that really all the experts, the physicians, the scientists are available at a specific time, and that they're showing up. I mean, it doesn't help if the bioinformatician is just written on the tumor board and he is a member, but he never shows up. Then you cannot get the information, because the patients are discussed in the tumor board instantly, and then later on it's difficult to get, you cannot make any more recommendation, a decision from the tumor board, because the members are not together anymore. Of course, you can ask somebody, but it's more difficult. That's why we are coming together in the tumor board and taking a decision, a recommendation in the tumor board. And I think this is the most critical thing. How many patients you are discussing, I think is not so important. If it's just 2 or 3, perfectly fine.
Well, it's not about the patient number, but the knowledge you can, the expertise you can.
Of course, I mean, this is another topic, and if there are specific databases for children, and you can connect to the Netherlands, maybe, then you have to look. If there's a database, then it's more easy. Then you can connect directly to the database. If this is a public database. If not, of course, then you have maybe to invite somebody, or then you need a recommendation, a second opinion from another tumor board. So this could also be an option to send to have a second opinion from another tumor board.
Thank you. Professor Leia, please. Go ahead.
Yeah, thanks indeed. Great presentation. I think we could have multiple questions to address and to clarify, but actually I'll start with 2 of them at least. So first thing, I think, and you partly answered already with your last slides, but still, what is the right approach to go? Because many hospitals, cancer hospitals, are in the process of developing electronic health records, those that do not have them at the moment in Latvia, at least, in the way that we anticipate. And in the case, if those are 2 different systems, the electronic health record and the tumor board system, then of course it's quite complicated how they can communicate between themselves. Or the other way, what you showed, that you are starting from the tumor board soft and then going maybe to electronic health record. What should be the right way to go with this?
Actually, I think this is not a big topic, because once the data are in a structured way in a database, You simply can transfer the data into different fields with different annotations. I think the biggest problem today is that most of the data are just PDF'd and sitting somewhere in the PDF, and those data are not available anymore for further analysis. But once the data are in a database, you easily can look for the mutation, for example, if you want to know if this is pathogenic mutation, yes or no. And you have the same field in the electronic patient report. It doesn't matter, because then you can simply transfer the data into another database, if it's already in a structured way available. If not, then you cannot, and this is mainly the problem. So therefore, I think it doesn't matter if you do it this way or that way. And the clinical database we are creating now is based on SNOMED code. That means everything is coded already behind, and you can easily even set up the database in, we have done it now in English, you can do it in French, in Chinese, whatever, because the code is the same. And you simply need the synonym from the SNOMED code, and you can easily translate into Spanish. And therefore, I think once you have structured data, problems are solvable. If you have no structured data, as we do it today, I'm always joking and saying, even in our hospital, we don't know how many lung cancer patients are currently treated. Maybe the financial department knows when they're sending out the invoices, but the oncologists don't know. And this is currently our problem, that everything sits somewhere, but you cannot retrieve the data. But once it's in a structured way stored somewhere, you need an informatician to maybe transfer the data to some other databases, but then it's solved, it's possible.
I think that's another important message, because whether to implement SNOMED CT or not, that has been from time to time discussed during the last 10 years, I think, in Latvia. Right. The other question, actually, what I wanted, if I may, is that as we saw, I think the soft was in English, so, but it is developed only for your institution, for your cancer center, right? Are you offering this also to be used, for instance, for the molecular tumor boards in Latvia? Could this be adapted? Thanks.
We can share. We are a public university, and of course we can share. Maybe we need a little bit of money for adapting, so that when we have to program it, but or for special needs. But in general, it's a public software, and we already have published it on the website, and everybody is free to use it. So of course, we are not a company who want to make profit with it.
So unfortunately, we need to move forward. So thank you, Martin. And by this, we move to the next speaker. And it's my pleasure to introduce Ed Schuring. Ed is professor and senior clinical scientist in molecular pathology at the University Medical Center in Groningen in Netherlands. And his research focused on identification of prognostic, predictive, and epigenetic and molecular markers for clinical outcome, response, and chemoradiotherapy, and gene-targeted therapy and treatment resistance in different cancers. My personal thanks to Ed is to be very responsive and open to share his knowledge already before with his team. We organized several Zoom sessions where he kindly shared his experience with molecular tumor boards. And his talk will be on a Dutch experience using predictive profiling to guide targeted therapy in cancer patients, role of molecular tumor boards. Floor is yours, Ed.
Thank you very much. The introduction takes already a lot of time, so thank you very much for that. Thanks for the invitation to be here today, and happy with the first speaker. Already introducing a little bit. I'll show you the way we do it in the Netherlands is a little bit different, and I'm going to share your experience. And it's a flight through a lot of data in the 15 minutes. So here are my disclosures for today. So in the Netherlands, for molecular diagnostics, the drugs that are available for targeted therapy is listed over here. You see there's quite a lot of different drugs that are coming into the field. And for that, we have to test more than 14 different predictive markers when we get a request for testing, for predictive testing. And this list is growing, and everybody starts with lung cancer, because in lung cancer everything started to develop, and the developments are quick. But on the other hand, you should also realize that all the questions I get from the other departments for testing, predictive testing in other malignancies. And this is not a complete list, but this is just showing that this field is moving very quickly based on all the drugs, targeted drugs that are becoming available. So there's a growing list of markers. So everybody started with single gene testing a couple of years ago. We switched to next-generation sequencing approaches with 33 genes, but recently we decided to move into the larger, broader gene test. And we decided to go for the TSO 500, which covers more than 523 genes. And many different bigger hospitals in the Netherlands are going in that direction. And the idea is that this will help us not to analyze for every patient the 523 markers for mutations, for copy number variations, tumor mutational burden, all that kind of abnormalities that are needed to make clinical decision making. This is all in this panel, but we, for lung cancer, for instance, define that we look at only 14 genes. And for the markers, we also define what to look at. And realizing that next year, when a new drug comes into the field, we have this workflow already running, and we just extend the mask how we look at the genes. So I think these kind of approach, these kind of developments help us to help us ready for the future. Now, to show you how diagnostics interpretation works, I would like to share a paper that we published last year. We have a pathology registry in the Netherlands already for more than 30 years. So every tumor, every tissue that's analyzed is in the Netherlands is in that database, in this registry. And from that, we took data from 2013, 15, and 17, and searching for stage 4, non-squamous, lung cancer, and EGFR. And we found in these 3 years more than 10,000 patients tested. And were found, and only 77% was tested. So this the first message already is that we are not testing everybody. And still, even the more recent data show that 20% of this group is not tested, while there is testing possibilities and drugs are available. But for today, I would like to share that in this group, we looked for mutations that were reported by the different laboratories in the Netherlands, and it turns out that there were about 11.7% of the data of mutations found in EGFR, variants found. Now, it's impossible to read this. This is one of the 3 pages of the slide, of the table that we published in the paper, showing all the different variants that we identified that were reported. And there are the common ones, everybody knows, but you can see many of the variants here on this list are only found 1 or twice. How to deal with these markers? Should we just not consider them, or what should we do? So the idea is, so if I summarize this percentage of uncommon rare mutations is 90% in this population. And so the question now arises how to deal with it. So what we did, we reclassified all the mutations, all the unique uncommon mutations using the international classification of the EMP-CAP ASCO guidelines. And then we looked, based on this information on pathogenicity and actionability, how the interpretation was done. And for that, we could couple our database to the Dutch Cancer Registry. So we had the information, whether the information we gave to the doctors, the patients were indeed treated with the drug or not, and what the response is. So this first slide shows you, in the left 2 bars show that of the cases of the mutations that are targetable, and about 80% of the cases, the patients indeed were targeted. So 20% not, although there was a mutation. But even more interesting, on the right-hand side, we have the resistant mutations, we know, and those that turned out to be not targetable, not actionable. And still, a quarter of these patients was treated with, by the doctors. So the information that people have at the moment is not complete because of the difficult analysis. So this is a survival curve from the patients that were treated based in this population. And the left 2 curves, the worst survival curves were, of course, those that should not have been treated. So classification, correct classification, interpretation is essential. So how do we do that in the Netherlands? A little bit different as we do it as we heard from the Swiss situation. I'm a clinical scientist in molecular pathology. I'm not a pathologist. I'm working already for more than 30 years in pathology department. It's an official training for 2 years. And we are responsible for performing the testing, molecular testing, choosing the assays, but also the interpretation. So in fact, and it's summarized over here, if we have a common mutation, a known mutation, we just report it in this pathology registry, and the advice to treat is simple because it's common. But in all those cases where we have uncommon or rare mutations, we have to go through a process, as you can read on this slide, to look for pathogenicity and look for actionability. And this is all done as soon as we get the next-generation sequencing data, and we, as clinical scientists, then define what mutations, what variants are difficult and should be discussed in more detail. So that's the difference how many countries work at the moment. So then we use, there is a lot of information, databases, tools, and there is no guidelines at the moment. We saw that we are all working on this, how to do this, but we have to see what is the choice. So what we do is, and there's an example here for an ALK-resistant mutation that came up a couple of years ago. There were a couple of them described in the middle picture. You see these mutations. And on the other hand, we had a lot of drugs coming up. So the question now is, what is the best drug you should give here? And for that, we're not only going on the left-hand side to all the information and tools that are there, but we're also building models based on data that have been published using these mutations and combined drugs. That's on the right-hand side. So there's clinical information, but this is a dynamic list. It's growing, and the data is quite small. For ALK and some other examples, in the middle, you see models, cell lines that have been transfected with these mutations, and then the cell lines in vitro biological responses measured with these drugs, whether they die sooner or not, or becoming resistant. So there's also information that can help. And as we saw on the previous slide, we're also doing some 3D modeling in the Netherlands. I don't have time to go detail, but the 3D modeling, as you see here, done by structural biologists in the molecular tumor board, is something that since we use the small molecules in these structures, of these virtual structures of the protein of the specific area we are interested in, with and without the specific mutations, and that helps us not only seeing whether it fits in, but really how it behaves in this three-dimensional structure. And that has helped us a lot, in addition to all the other information, to make decisions on what drug of these lists fits best with this mutation. And this is just to illustrate the response of patients treated with ALK-resistant mutations. That all the decisions we make every time the tumor became back to the hospital with resistance, we sequenced and we found a new mutation, that we now can extend in this population survival in this fatal disease for, in most of the cases, a year, but some of them, more than half of them, for many years, giving them a good life. So this is just an example of how it works. So what do we bring into the tumor board? All these, not only rare mutations and common mutations, but also those that were found in other diseases, malignancies, those that do not have standard care. And this, so there's a lot of, so we are not bringing in the common, it's only the very exceptional complex data. Now, the tumor board is similar as we showed previously. We started off with a pulmonologist, pathologist, and a clinical scientist to be present at all time. We have also a guideline in the Netherlands that defines that they should be in and what should be done. But the more recent, the meetings we have every week now also includes medical oncologists, clinical geneticists, structural biologists, bioinformatics, and all these people are there, and they are experts. And expert means it's not a pulmonologist that's there, but the pulmonologist, the expert in the MTB in the Netherlands, should be a doctor that really treating patients, many patients with targeted immunotherapy. So the experience is there. Now, looking a little bit what we're doing. So in 2018, we discussed mainly lung cancer patients in blue, and then you see in time that we are switching to other diseases, to other malignancies discussed in the molecular tumor board. And the changes are coming up because we had a new guideline was introduced for lung cancer, and we see in 2020 more cases. And then in last year we became reimbursed in the Netherlands, and then suddenly you see a lot of CUP cases coming up in the tumor board. So this is the field that is moving, so you need other and more experts. Does it work? Now, so in 2018, all the cases that are discussed, we analyzed them, we collected response data after a couple of years, and then we found that the overall response rates for those cases that were discussed in the molecular tumor board was 67%. And this is quite good. That's better than phase 1 studies, indicating that it's really worthwhile to discuss these cases in molecular tumor boards. In the Netherlands, we have not only in Groningen, but we have 8 tumor boards working now, molecular tumor boards, all in the tertiary referral centers. And until now, lung cancer is the most common, but we see that other malignancies are going to be discussed. So we did a study where we compared by external quality control the interpretation of complex variants between these 8 laboratories, and the concordance and the inter-rate reliability was really very high. And this is important to do this, because everybody has his own way of working, but by controlling this by difficult samples, we learn and harmonize how to work and give similar results to the patients. Now then, of course, we have, and all this hospital, I'm not a doctor, but in hospitals, every hospital has his multidisciplinary tumor board. And that is not the same as the molecular tumor board as we're discussing this morning. Because I don't have time to go to all the data here, but importantly is the casuistry, the cases that are discussed are only the complex and the rare cases in the molecular tumor board. And we also look for more data, scientific literature, profiling, etc. And most importantly, we spent about 15 minutes on average for each patient every week. And so we discussed only a few cases, while in the multidisciplinary meeting, all patients are discussed. And I guess you as doctors know that it sometimes takes less than 1 minute to discuss them. Looking to our region, we have 16 hospitals, and it's quite important to have a good interaction between the doctors. I can't say it works, but we have to work on this, because the information that we're getting and the information that we need, the harmonization of that is very difficult. The field is moving so quickly that the area we're working on is very difficult. So harmonization between all the hospitals and the molecular tumor boards, the central molecular tumor board, is very important. That means also in the Dutch situation, as I mentioned, we also need to collaborate. So we're building here a knowledge center that brings together and harmonizes all the activities that are happening. And the Netherlands now, these molecular tumor boards has come to a situation that, regulation authorities, health authorities now, are using the molecular tumor boards as the central molecular diagnostic laboratory to help them making guidelines in the Netherlands, trying to organize now that reimbursement of very expensive, complex testing will only be done through cases that were discussed in the molecular tumor board. So really the molecular tumor board is becoming the leader in molecular diagnostics in the near future. But don't have time to do that, but we heard in the European meeting, we heard a lot of experiences in Europe, but we need also to hand out and share this information, not only working, but also the data that we see with each other. Because if we have that very uncommon mutation in Groningen that we saw, there will be somebody else in the world eventually also having this mutation. So when we build a database with these molecular tumor board cases, in the end, it will not be rare variants anymore, because we can share this information and experience how to treat. So we have to really, there are a lot of initiatives going on, but we should work on this very hard. So nearly to the end now, showing on the left-hand side, the molecular tumor board today. It's small, it's few specialists, it's not regionally built yet. The data are based mostly on lung and cancer, but the near future is, on the right-hand side, that it needs to be structurized. So it needs to be networking building. It needs to have much more people involved as we're having now, because the complexity of the data and the different diseases that come up in the global are very large. Now the last slide is the take-home message slide, is more, this is the Dutch molecular tumor board model that we believe. So the molecular tumor board should have experts that is different from what we do in the multidisciplinary team meetings. As I mentioned, you can read it. It's focusing on rare and complex mutations, so we do not discuss in the molecular tumor board all the other, more than 90% of the other mutations that we found. It should be located in a central touch cancer reference center, open for easy access, and we do that by video conference. On the same reason, by COVID has helped us there, that this meeting is totally now by video conference and making it very easy to join everyone. And as I said, the lead in cancer. Now, because of time, I leave it here. Thank you for your introduction. Of course, a lot of different groups and departments and institutions were involved in this. In Groningen, we were really the leading to get these molecular tumor boards at the national level in the next step, but I'm not finished, because there's a lot of work to do. I'm looking forward to other talks today in this session on molecular tumor boards. Thank you very much.
Thank you very much for this very nice overview, and I think you're even more advanced than Switzerland in terms of data sharing, because we do not have any access to the data from Geneva or Lausanne or Basel when we are working in Zurich. So we have directly to contact the physicians to ask for their expertise. One question I have, you have shown all these different models when you do the modeling for drugs into the domain. How you present this data in a tumor board? Because typically a patient is discussed 5 to 10 minutes. I don't know how you do it in the Netherlands, but no. we really have only a short amount of time when we are discussing this in a tumor board. So how do you present these data that an immediate decision can be taken by an oncologist for the treatment?
Yeah, different from what you are doing. It looks like that in you using your molecular tumor board to do a lot of these analysis using your database or your tool. So we prepare, a team prepares the molecular tumor board meeting beforehand. So if we go into the molecular tumor board, these models were already generated, the conclusions were drawn, and then of course that makes it exciting to come to a molecular tumor board, and the structural biologist shows these nice pictures moving and explains, and that's very hard to understand, explains why this drug is better than the other. But the analysis is already been done, and that accounts for all the analysis that we do. So we prepare, the clinical molecular scientists and molecular pathology, prepare all the cases for the molecular tumor board, so we have spent the time knowing the background of the patient, and the doctors that know what clinical trials are out there, the experiences of others. That's what we really do in the molecular tumor board.
Okay, thank you. Alvin?
Thanks for the great talk. So did I understand that correctly? So you have a nationwide database that includes sequencing data of every single patient that is sequenced in the Netherlands?
Yes.
And you are able to connect that to treatment and outcome data for those patients?
Yes and no, because the cancer registry database is another database.
Okay.
And linking all these databases is a lot of people are working on this to get this, so it's not actually available, but we can find the information. In fact, in the molecular tumor board, the registry, cancer registry is behind, so the data are not new, and we're discussing patients treated today. So we need really the clinicians that treating the patients in the tumor board, we invite them, because they know the information. But this is more for searching and research, that you look for the diagnosis. Yeah. So in the pathology registry, we only, by the way, it's not research based. So we only put in molecular data, pathology data, electron microscopy, immunohistochemistry, everything is in there for the purpose of doing diagnostics.
And can you, like in a few sentences, explain who set up that database, who was in charge and who managed to do so? I mean the sequencing database.
Now, the sequencing database was added in this registry later because the registry was already there. And we, as a clinical molecular biologists in the Netherlands, sit together with pathologists and the group who is responsible for building this registry to see how we organize that. So we build a module. We are now building a module where you have to fill in all the steps that you do in molecular diagnostics, and then it's standardizedly stored. And recently, we're also now putting in the VCM and the BAM files. Personally, I don't believe it's a good idea, because I did the analysis already, did all this difficult analysis, and we don't want people to start reanalyzing this, because there will be a VUS might be not a VUS for somebody else, by the way.
Okay, but that was an academic endeavor, so you sat together and planned to do so.
The pathology society is in that, yeah. We can have a chat afterwards, because it's...
Thanks.
Okay, thank you very much for this very nice overview and the way how the Netherlands have solved the problem of coming along with all the data. Thank you. Now coming to our next speaker, it's Karolis Saplauskas. There he is coming. Karolis is a hematologist at the Vilnius University Hospital, Santaros Clinic, and is working on diagnostics of inherited hematological diseases. And his research deep applications are deep learning, next generation sequencing, and de novo CNN. Neural networking, I think, is one of his topics he is in research. And the talk he is giving today oops, I'm sorry, is personalized treatment of acute myeloid leukemia experienced of Vilnius University Hospital Santaros Clinic. Thank you very much, and we are looking forward to your news.
Okay. Thank you very much for the introduction. I'll try to keep it brief, and it's an honor to be speaking today, to be wrapping up this session. And the organizers of the event asked me to show an example of how we can start with molecular tumor boards or personalizing the treatment in a country that's very similar to Latvia. So here you can see Santaros Klinikos, and we are located over here. So the campus is constantly expanding, and actually we recently had to build these 2 new floors. So just a single slide to boast on some numbers. So here you can see the rates of allogeneic stem cell transplants in Europe. And as you can see, Lithuania is over here, and we are one of the leaders in the region in terms of numbers of allogeneic stem cell transplants that we're doing. So leukemias and transplants is one of our core competencies. So that's actually the difference of us to other centers that we heard about today, is that we started not from lung cancer, but from leukemia. And I think one of the take-home messages is to start where you're already comfortable. So I will have a different perspective compared to other speakers. Instead of focusing on the nuts and bolts of of the function of molecular tumor boards, but how we got there in the first place. And there's different ways to implement them and to run them, but I think the #1 step is to set it up. And for us, it was really the search for new treatment options for some of the patients, as well as trying to keep up with advances in genomics. So just briefly, acute myeloid leukemia is a cancer that in general originates from these immature cells, and the problem is that these cells don't allow for other cells in the blood to function properly, and this can lead to all sorts of problems. And it's a really aggressive cancer, and it requires intensive treatment. So part 1 is searching for new treatment options. So to sum up the pathway that the patient goes through once they have the diagnosis of acute myeloid leukemia, they usually receive induction treatment and consolidation, and around 60% of your patients will go into remission. Unfortunately, there is a fraction of patients that have refractory disease. They don't respond to the first line, and then there are other patients who will relapse. So this is a setting known as relapse refractory acute myeloid leukemia, and you can understand that it's a challenging situation to treat, and it generally has a poor prognosis. So this was the setting that we decided to focus on and to try and establish our molecular tumor board. And one of the options that was commonly used for relapse refractory leukemia is called FLAG-IDA, which is a very intensive treatment, and it has a high level of toxicity. So we had a goal of finding a safer and lower toxicity alternative to this regimen. So essentially we looked at the literature and decided to focus on a combination of these 3 drugs as a backbone, and then trying to personalize that to each specific patient. So in our center, this is called the active protocol. Essentially, the patient goes through 2 induction cycles, and if possible, we try to bridge them to allogeneic stem cell transplant, or if that's not possible, we can continue with maintenance therapy. Right, so this was the clinical part of the story. basically trying to solve one of the problems that we encounter. The other side of the story is trying to deal with all the genomic information that's coming up. And I will show the results of that protocol later on. So when it comes to analyzing genetic information in AML, I would structure the tests in different tiers. So we have very urgent tests where we need information ideally in a day. And then we have other tests where we have more time. So this includes SNP arrays, targeted NGS, RNA-seq, and so on. Additional layer is germline variants, and in that case we're using fibroblasts because we cannot use blood, and in the leukemia case you would just be sequencing cancer. The point here is that actually establishing the operations for doing sequencing from somatics, the bone marrow, and also the germline was one of the challenges that we had to solve because it takes time for people to get used of doing those sponge biopsies, sending fibroblasts to the lab, and actually ensuring the quality that you can extract the DNA from fibroblasts. But essentially, we want to look at the germline variants, so perform whole exome sequencing for these patients. And one of the questions that we had was, all right, so which tests should I order when? And one of my key messages here is that it's better to keep it simple and to standardize, maybe sometimes to do a bit more than necessary, but the less if-then statements you have into your protocol, the higher the chance that people will actually be implementing that. So we decided to screen all patients who are eligible for allogeneic stem plant and are diagnosed with acute myeloid leukemia. And you might be asking the reasoning for that. So what we saw and what is known from literature, that even if you sequence all of the patients, regardless of their age, around 10 to 12% of cases will have hereditary predisposition, which in our opinion was significant. Knowing this information affects your choice of donor. So if there's a germline variant and you have a sibling that's a potential donor, you would want to evaluate them as well. Then there is the research perspective, but again, I would like to stress that the operational benefits when you have a simple protocol that you include almost all of your patients in this analysis makes things easier. So once again, if we have a diagnosis of AML and the patient is transplant eligible, which generally means that they're fit and they're 17 years and younger, we take the skin biopsy and perform whole exome sequencing. Right, so once we have all that data, the need for molecular tumor board arises naturally because you have more information than you can deal with. So just to briefly summarize how we structure it in Santaros Clinicos, since we're working with leukemia, the pathologist in this particular setting is optional, and we invite them for the cases where we have some specific questions. But generally, we do it similarly to the Dutch way, where we ask every specialist to prepare their part, to do their homework, and then to present the information. And yeah, I think one of the messages today is that you can have different implementations of molecular tumor boards. In my opinion, the most important part is to get started and to build expertise. And one of the differences to the Dutch way is that at the moment, we try to pass all of the patients who are in relapse refractory setting through a molecular tumor board. The simple reason is that we want to build experience. And it generally takes us from 5 to 10 minutes to reach a conclusion. So once again, we have two parts. One is the clinical need, and the other is the molecular information that's coming. And this can help you to decide when you're in an unknown situation. But another benefit that we saw is that you can see some patterns and then put them into reusable protocols. So hopefully, as you see more cases, you don't need to start from zero. And this is actually something that we implemented in Santaros Klinikos. So this is active protocol, but as you can see now, it's specialized based on different abnormalities that we detect. And one of the last slides is, these are the results from the active protocol. So this is a retrospective trial where we compare active versus FLAG-IDA. And of course, there's bias in that. However, the patient demographics are pretty similar. And the only thing that you need to know from this is that these lines over here are higher than the previous standard, which is FLAG-IDA. So we show that there's better survival, at least from what we can see now in the active group. And the number one objective that we had was reducing toxicity, which we were able to achieve. One final slide. One of the interesting things that we saw once we started with the active protocol is that you can, in the beginning of the treatment, see which patients will respond. So on the x-axis, you have days in the treatment cycle, and just to show you one example, this line over here, which means that in 4 days, you start from almost 100% blasts in the blood, you go to almost 0. And we can see that you can give a good indication who will respond to the treatment. So what we've learned so far, that it takes time and effort to become consistent and to really implement this, but reducing operational complexity and continuing on educating your colleagues appears to be key. So thank you very much, and I will be happy to answer your questions.